de novo protein sequencing algorithm is not very effective when we use it against the known and unknown proteins, but it's very useful when we use it against the different types of spectra that is almost complete or near to completion. And as we know that the different types of spectra, they are almost incomplete, so we can get the very ambiguous solution for these spectra. If we had access to a database of all proteins from a genome, then we would no longer need to consider 22 L peptides, that we know that we have to generate different types of peptides with different sizes. And we sequence to interpret an MS-MS spectrum, but could instead limit our search to peptides present in this database. Now the approach to find and identify the specific peptide or the protein is not to generate different types of peptides by using MS, but the new technique is to search the sequence of the protein in already present databases. As for example, if we want to find the answer of any solution that is present in the textbook, we just find the answer in the last pages of the textbook. So similarly, we just compare the sequence of these peptides that are present in the databases. So the best solution means the best sequence of the peptide will be that matches with the peptide that is present in the database. And on working on this algorithm, algorithm known as sequest is developed by the John Yates and its colleagues, and the function of that algorithm was to find the best possible sequence that is generated from the experimental data and compare it with the already present sequences of the proteins in the databases. So we formulate the protein identification problem as follows, and we know that we have to find the protein from a database, as we already discussed, that best matches the experimental spectrum. So we have to find the experimental spectrum in the database. So for this algorithm, the input will be a database of proteins and experimental spectrum S, a set of ion types, and a parent mass M. So the output will be a protein of mass M from the database with the best match to the spectrum. So it will be much easier to find the sequence of the protein by using the spectra that is generated by MS and comparing it with the sequences that are already present in the databases of proteins.